Hi everyone. I know we like to concentrate on our Meet the Scientist series, but I think it's also important to meet both the graduate students and the postdocs that make this work possible. And so I wanted to introduce you to two graduate students. They're in the lab of Professor Jill Banfield from UC Berkeley. We have Jacob West-Roberts on the left and Susan Mullen on the right. And so they're both going to walk us through what they're up to today and tell us a little bit about the sampling that they're doing in a long-standing geochemical well that we've used. That's called the Shumway well, named after the owner of this cabin, the Shumway cabin. It's Scott Shumway. He's graciously allowed us to sample his well. So Susan, why don't you explain to us what you're doing and why you're sampling groundwater from this well?
Sure. So we're really interested in microbes that can't be cultured, and what we do is we take a pump and we flow through a lot of water through these different filters. So this setup is called a brewery, and we have 4 different contraptions that can hold filters. And right now we're just doing a bulk filtration, so we'll capture most of the community with a 0.1 micron filter. So anything smaller than that, which would be like really small viruses, will come out the other end in the filtrate, but we'll capture most things. And since there are 4, we can also have larger sizes, and we could do a size fractionation to get all the different size microbes. And yeah, we just use this peristaltic pump, and we pump the water from the well through here, and then it just comes out, and we then will freeze the filters and extract the DNA and find out what's living in here.
Excellent. And so we're trying to tie into the same depth below land surface that we have a long record of geochemical data. So they're right now pumping from a depth of roughly 150 feet below land surface. I will note, however, that a lot of these wells were not specifically developed for drinking water purposes, and so as a result they don't have particularly high flow rates. And so Susan and Jacob have noticed that some of the flows emanating from these wells that we had hoped to sample throughout the watershed are producing at a low enough flow rate that it's going to be a little bit challenging until we come up with another creative set of component. Jacob, why don't you introduce yourself and sort of give us an idea as to what you've been interested in and are currently working on in Jill's lab?
Hi, so I'm Jacob. I primarily do bioinformatics and computational stuff. Right now I'm working on some metatranscriptomic data that a former postdoc in our lab, took from the hill slope over at the pump house. So we're looking there for lots of signatures of selenite, arsenate, selenium and arsenic species, metabolism and transformation in the soil, because there's a lot of it over there.
And those are soils built up on shale bedrock. Of course, it's the pump house. And that would be a nice opportunity to tie into this particular well, which exists and is completed throughout the same Mancos shale formation, albeit a little bit higher in the stratigraphic profile relative to pump house. Well, thank you both. It's a beautiful day to be out here working, and good luck.
Thank you.
Thank you.